hospital. Okay, we're going to jump around kind of all over the place here. I just want to do a quick review of everything we've talked about so far. Okay, so we're really going to literally jump all over the place. So go ahead and jot some notes down if you like. It'd be a really good idea because you can use this stuff on the test to help you out. Okay, so first let's just dive into Punnett Squares. Okay, if there's anything that confuses you, just stop me and ask me. Okay, so everybody I think is familiar at this point how to do a Punnett Square. We make the box, right? And if we're going to cross two parents and maybe one's purple and one's yellow, right? And you can tell up here the yellow is recessive, homozygous recessive, it's the little allele, and we've got a homozygous dominant one up here, right? The big piece. So it doesn't matter what letters we choose, usually we just kind of represent the dominant one with the word, the first letter and its word, whatever it happens to be. But it doesn't have to be, right? Then we separate the alleles. In other words, this parent can give a big P or a big P, right? So we've got to have two spots there for it. This parent can give a little p or a little p, so we've got, actually that one's over here, we've got two spots for little p, two spots for the big P. We do the cross, every single one of the babies is heterozygous, remember that word? So they have a big and a little. Then we sometimes have to work out genotype and phenotype frequencies. And you're going to see this on the test, I know you've done problems with it already. So the F1 is the filial one, that's the first babies, right? So what I'm talking about when I say what's the genotype and phenotype of the F1, I'm not asking about the parents, I'm asking about what happened in here, okay? So we're asking for probabilities. Genotype is how the genes are. Well, 100% of them are big P, little p, okay? Phenotype is how they look. 100% of these things are purple, okay? Just like that parent, only they've got different genes, don't they? And that's how you do genotype, phenotype frequency. It's really that easy, right? If you're confused about how to add these up, remember, think of a dollar bill. There's 4 quarters in a dollar, right? So if I've got 2 of these, that's 50%, right? If I've got 3 of them, it's 75. If I've got 1 of them, it's 25. So pretty easy to remember. Now let's say that we cross these F1s, because you're going to see this again on the test, I'm sure. If we take and cross an F1 generation, we take 2 of the babies, which is a little creepy, but it's totally cool because they're plants. They can do that. It's all right. Nobody gets up and out of shape. So we're going to make 2 babies. And that's going to be what we call the F2 generation. Okay? So we're going to take these F1s, purple and purple. They're big P, little p, and big P, little p. So when we make them, there we go. We're going to end up with, we've got to separate the alleles again, right? Because each parent can only give 1. So big P, little p, big P, little p. We do our cross like you guys know how to do. But now when we figure out our ratios, I need to write all those down. We've got a couple of different genotypes, don't we? We've got big P, big P, big P, little p, little p, little p, and some different distributions. We've got different phenotypes. 3 of them are purple, 1 of them's yellow. Purple, 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 and we've got a yellow one over here. Probably should color those the other way, right? If we want to work out percentages, 75% will have the phenotype purple, 2, 3. Okay? That's also, you could write it as a 3-to-1 ratio. You might see either or on an assignment or the test, because it's 3 to 1, get it? Okay? And if you were to take 3 and divide by the 4 that you've got, you get 75%, which most folks can recognize how to count that up though, right? And we can do the same thing for genotype, right? We could figure out the frequencies for genotypes. Okay? Big P, 25%. Big P, little p, 50%. Little p, 25%. Simple stuff, right? Hopefully that's like really super review for you there. Okay, let's move on to some kind of more new-ish stuff. Let's compare both, a bunch of stuff up here, both meiosis and mitosis. We have not talked about meiosis yet. We're going to real soon here. Mitosis we have talked about. Do you guys remember IP, Matt? Where we did interphase, prophase, metaphase, telophase, right? All that stuff, the anaphase, all that stuff. So mitosis is just regular old nucleus division. The nucleus of the cell copies the chromosomes, divides, boom, you got 2 new cells. Cell cycle. We did that, went down to lab, we counted them, like it was like, we did a lot with that. Okay. this is what your cells do to make copies. Well, if you're going to make a copy of a cell, you better have all the same number of chromosomes that the original cell started with, right? And so, what we call these cells, we call them diploid. They have, sometimes we write it 2N. They have 2 sets of chromosomes. Why do they have 2? Because they got one from mommy and one from daddy. So for every characteristic you have in your bodies, you have 2 versions of it. Because one came from mom, one came from dad. So the babies, the fully grown humans, or whatever we're talking about, plants, have got, all their cells are 2N. They're diploid. Okay? And for us, we have 46 chromosomes. This is that whole cycle that we did. Over here, meiosis, which sounds really similar, is basically mitosis twice. So think about this. You need to get half of the DNA from one copy of the DNA from dad, one copy of the DNA from mom. How the heck do you do that without doubling everything up? Without having, 2 times the DNA? So what you've got to do is have it out a little bit, right? So what you're going to have to do is undergo mitosis kind of 2 times. And what's going to happen is we're going to make sex cells that we call gametes. So sperm and eggs. In order to do that, the cell undergoes that nuclear division sort of thing, where the chromosomes all spread out and copy each other, right? They line up in the middle in that whole metaphase looking thing, and they pull to either side into the cell. Then that happens again, okay? But in this process, what we end up doing is dividing up these chromosomes so that we have 4 cells when we're done, rather than the 2 we had over here. We started with 1, we ended up with 2. Over here, we're going to end up with 4 when we're done. Okay? And all of those 4 cells will only be 1N. In other words, they'll have half the chromosomes that did over there. See? So they've got 23 chromosomes each. And this works because we're going to sperm meets an egg. One carries 23, the other carries 23, and we're left with 46 when they get together. How about that? Works out just fine, right? So this is basically 2 rounds of this cellular division sort of thing. It starts with an interphase and it kind of goes through this twice. I'm not going to go through the whole process today. This is just a real quick, dirty overview. Okay? So what if I told you, just as a, try and work this out and understand it, what if a haploid sperm, it's a sperm, had 10 chromosomes? Okay? When it creates a diploid creature, how many chromosomes is that thing going to have? If the sperm has 10, when it unites with an egg, how many is it going to have? It's real hard math. If it's a sperm and it has 10 chromosomes, when it finds an egg and they get together, what do we got? 20. It's real hard math here, okay? Don't miss a question like that on the test. Super, super easy, right? The sperm has half. When the egg has the other half, when they get together, they've got, you add it together, you got 20, okay? In the case of humans, that's going to be 46. It'll be 23 and then 46 when they're together. But that's basically the gist of mitosis versus meiosis. But the words look really similar, don't they? Okay, so be careful with those. Okay. Let's talk about this thing called the independent assortment that we did. It relates to this. It relates to meiosis, okay? So, for instance, if I'm just talking about X's and Y's, that last set of chromosomes, when a female makes eggs, for each round of this meiosis stuff, you're going to make 4 eggs, okay? And so every single egg, if we're talking about just that last set of chromosomes, is going to get an X. Why? Because she's only got 2 X's. In the case of sperm, when a boy makes sperm, he's going to make 4 sperm every time we go through this meiosis thing. Okay? Each sperm is going to have either an X or a Y, but in a 50-50 ratio. So 2 are going to have X, 2 are going to have Y. That's why we end up with 50-50. Every time that there's a baby born, it's a 50-50 probability it'll be a boy or a girl. Okay? Because this is why it divides up right here. And I wrote your independent assortment in gamete formation, which is the eggs and the sperm. genes for different traits separate. Okay? Genes for different traits separate. And we're going to inherit one from each parent. They'll come back together. Let's look at another thing that happens during meiosis. But you there? I don't want to go too fast. Let me know if I'm going too fast. Give me a second. Slide this up and do this. So, these look familiar now. Remember mitosis, where we had the X's, right? The chromosomes, they line up in the middle, do their metaphase thing, anaphase, they pull back, okay? It looks kind of similar in a lot of ways to meiosis. In meiosis, another weird step happens. Okay, this is a meiosis thing. When we're making sex cells, the whole point of sexual reproduction has to do with shuffling the DNA. Has to do with baby gets a mixture from mom and dad. And that's evolutionarily very beneficial. Why? Because when you mix stuff up, you maybe provide resistance to diseases you've never even met before. You have different characteristics that come about in the population. Evolution can act on those things as long as the DNA gets shuffled around. Well, just getting half from mom and half from dad is not that much shuffling. So what your cells do is when they make sperm and egg, they do this additional thing called crossing over. So when all those X's line up in the middle, okay, instead of just being perfect copies and spreading apart to the other ends of the cell, what they do is that some of those little chromatids cross over. Like you see, here's two sister chromatids. The A's here represent a gene, okay? The B's represent a gene. Maybe that's blonde hair, blue eyes, you know, whatever happens to be, okay? And then what happens is those two chromatids touch each other and then cross over. So they swap. It's almost like shuffling the deck. Now, why does blonde hair and blue eyes always go together for the most part? They just happen to be really close together on one of these. So if they ever get shuffled, they always go together. It doesn't have to be, but usually, right? So there's some weird genetics that happens. But this is called crossing over. That's really all you need to know. Happens during meiosis, and it's a further shuffling of the DNA. It's like the whole point of this sex stuff is to shuffle that DNA up. So we get a mixture, we get a different stuff, different characteristics, new characteristics, things that maybe have hidden a few generations, right? Or in different combinations come together in the babies. So those are called sister chromatids, okay? Parts of the chromosomes. So when you keep hearing me say chromosomes, I'm talking about this big package of DNA. And on the chromosome, there's individual genes. Like there's the gene for hair color. There's actually many genes for hair color, right? Gene for nose shape, gene for skin color. There's lots of genes for skin color. Right? All kinds of different genes, and they're on these in specific locations. Okay. All right. Let's talk about. Continue with a lot of stuff here, guys. I know, but get through some stuff here. Put that whole thing up there. So here are some different types of heredity that were different than what Mendel did, okay? Remember, Mendel just did big T, little t, dominant or recessive. But there's all these complex forms of genetics. There's stuff called multiple alleles, where you've got multiple versions of a gene. In like skin and hair color we talked about were examples of that. There's many different versions of the gene, so you can have such a huge array of different skin colors or hair colors or eye colors. That's when you see that big distribution. It's not just either blonde hair or brown hair. There's this humongous distribution, and that's why. Okay? In just regular old dominance, it would be that, right? There'd just be tall and short. There wouldn't be any in between if it was dominant type thing. Codominance were both got expressed. So everybody recognize what I'm saying when I say alleles? Alleles are versions of a gene, okay? So like you've got a big allele and a little allele, big T, little t. Those are the alleles, they represent genes. So there's a gene for tall pea plants, there's a gene for short pea plants, okay? Those are the alleles I'm talking about. In codominance, both of those get expressed. In other words, the short one doesn't get hidden by the big one. So they both get expressed, and we represent them as two big capital letters. And our example here was checkered chicken. In codominance, you don't get either black or white chicken. You get both those alleles. If the child has you know the big B and the big W allele, they're black and white. They're mixed like that. Okay. Brown cows, right? We got the cows that are brown with white spots because you made a brown cow and a white cow, and you get spotted children. Okay. Then we had incomplete dominance, which the difference between these two is that this is like a mix between the colors. So we had the white flowers, we had the pink flowers. When we mated them, we expected to either get white or pink, right? Or maybe we expected to get white we had the white and the red flowers, and when we mated them, we expected to get white or red, right? Or we might have expected to get white with red spots. We didn't get either of that. That would have been codominance. Instead, we got pink. So they blended. Really weird, right? So what happens is you get a mix between the colors. So the red and white alleles in peas give a heterozygous color pink. So the heterozygous ones, right, give you a pink one. So one allele's not totally dominant over the other. Both of them get expressed in a way. And last one, then we're going to do a couple examples here. Okay. I'll give you a second for copying that down. Good? Okay. That's a big set of notes here that I'm going to give, and then we're going to do an example. So the first thing I want to talk about are pedigrees, okay? And that's the new thing for today, is these pedigrees. So in a pedigree, we show generations. I know you've heard of like, oh, my dog's got a pedigree or whatever, right? Which kind of means a little bit different. It's talking about that we know the ancestry, the history of your dog, essentially, right? But in a pedigree, what we can do is trace characteristics as they move through the generations. So you're going to have, it's going to be separated into kind of lines like this that go across. You'll have a generation 1, if you're the parents, right? Generation 2, a generation 3, generation 4. Now, some of these generations include individuals from outside of the mating pair, okay? Like this one here is not at all related to the two original parents. See that? And in this scenario here, I was working out that we had a dominant, this is regular old dominance. How do I know it's not sex-linked? Remember, we just did this the other day. In a sex-linked characteristic, it's going to show up only in the boys, and we're going to see some females that are carriers, or more often in the boys are going to have the characteristic, and there's going to be some carrier females. How would I show a carrier female? Females are the circles. I'd have to shade half of it in, wouldn't I? There's no females that are half shaded in up here. So that should be an immediate clue that this is not sex-linked, okay? Because there'd be carrier females. I'd also see less females with it, and there are less females with it, but there are 2 who definitely have it here. Okay, so the characteristic we're talking about is they're colored in if they've got it. So what I did was I sorted out, I worked backwards to sort out that this one here, she had to be little h, and he had to be big H, little h. Okay? And as we worked on through, okay, we had this is a female, was their daughter, their son, their daughter. Okay. Now in this generation, she got it. She inherited that big H. Okay. He must have got big H or little h, and she must have got little h, Yeah, see how you can figure this out backwards? Now let's look at this. Okay. Do we know that that's big H, little h, or could it be big H, big H? We actually do, don't we? Because I gave you these, could have only gotten that big H there. Would not have got a big H from over here. See, I'm working the Punnett squares in my head. When this one mates with this one you know, don't you? Why do you know that genotype? You know the genotype of everything up here that's not positive for the trait. Because the only way to not be positive for this is to be little. So you can go little h, little h, little h, little h, little h, little, okay? Just like that. See how that works? Okay. Let's look at what happens in like colorblindness, which is X-linked. It is sex-linked. That's over here. And I kind of drew in what was going on. I colored it in red here. So you've got this X-linked trait. It's colorblindness. Who gets colorblindness more often, boys or girls? Boys do, okay? The reason is that boys only have one X chromosome in their last set. They're XY, and this is carried on the X chromosome. So I colored it in. This is a female, XX, and on part, she's got one of her X chromosomes, she's got colorblindness. This is mom. I colored it in red here. Is mom colorblind? No, she's a carrier, because she's got one good X chromosome that's making the appropriate proteins that you're not colorblind. But over here let's call it a broken one, okay? She's got a problem with this X chromosome that's going to lead to colorblindness. Over here, dad doesn't have it. XY doesn't have colorblindness. They mate, okay? And then we've got a situation here where none of their female babies have it. Their female babies can't have it, but 50% of their female babies are going to be carriers, like this girl here. What percentage of their boys are going to have it? 50%, right? Half of their boys, if it's a boy, is going to have it, and the other half is going to be free and clear. That's how sex-linked works. Okay. What if we, just goofing around, what if we mated a carrier female with a boy who is colorblind? What would the scenario be? It's like if you're colorblind, you could work this out and figure out what your parents are. It's kind of interesting, right? So let's say that the male's here, he's colorblind, the female's a carrier. Now all of a sudden we can have females that are colorblind. Okay? 50% of the females could be colorblind. You've got a carrier female, one boy that has it. Okay? If you're a boy that's got it, you had to get it from mom. There's no way dad could have gave it to you because dad can only give you a Y. See how that works out? So that's kind of how we sort out sex-linked diseases. Here's an example of a couple of sex-linked diseases, some famous ones here. Hemophilia, Fragile X disease, muscular dystrophy, those are all sex-linked, and we can trace them. Same way as this, okay, it's colorblindness. We're going to glance back at blood types again. We had a question over this, and it was confusing, yeah. And we were asking in the question, is there any way that you could have a baby with type O blood if your parents were type A and type B blood? And the answer was yes, you can. And I worked out a Punnett square. Okay? So there's multiple alleles at play here, and there's also codominance at play here. A lot of stuff going on. Let's say that mom here is type A blood. There's two ways to be type A. Okay? It could be IA, and little I. Okay? That's the recessive one. So if we separate mom's alleles like this, and dad's type B, we could grab this heterozygous type B for dad. When we do the Punnett square, 25% of the babies end up as type O blood. Pretty cool, huh? So you have AB parents and have type O blood because of those recessive alleles. And you're going to have one baby with AB, one baby here with B, one baby with A. How that Punnett square works out? And I just wrote this up here again. We've done this over and over and over. Alleles are just different forms of a gene. Okay? P stands for the parent generation. That's big T, little parent, little t, parent. F1 is their babies. F2 is what happens if we make two of their babies. We're only going to do an F2 if all the babies are the same, okay, for the most part, right? So if they were all big T, little t, then we'd probably do like an F2 to see. Okay. All right, that's a lot, guys. Last thing we're going to do here. And I'm going to let you guys work. Everybody still right? Everybody good? Okay. This is a pedigree. Okay? This is a pedigree. And I want to try and sort it out with you and see if we can figure this out. This is for photic sneezing. And photic sneezers are people who walk outside and see bright light and sneeze. Anybody ever do that? Walk outside in the sunlight and suddenly you sneeze? It's actually a genetic characteristic. And we can trace it. Pretty neat. Maybe you didn't know that. So we've got some generations here, right? So let's just label our generations here. We've got generation 1, generation 2, generation 3, generation 4. We have two sets of parents up here and a set of parents over there. Okay? And they're giving rise to some children. And look, their families cross over, don't they? Right here. This F mates with this E over here, and they start their own family over here. Okay. So we've got some questions here. We want to sort this out. It says here, to be a photic sneezer, that's a recessive trait, which we're going to say little r. To not be a photic sneezer, that's a dominant trait, big R. So, let me ask you this. Which genotype up here do you know without a doubt? Do you know for sure? Do you know what C is or do you know what D is? One of those, you can tell me the genotype. You do. What did you say? C. C, we don't know. Check it out. C could be one of two things. C could either be since this is dominant is non-photic sneezer, okay? We know that his phenotype is non, okay? So it could be, I'll reason R is big R, or big R, little r. See that? We know D for certain. There's only one way that this individual can be. She has to be little r, little r. It's the only way the recessive, which is why when we do a test cross, and this will be a question on the test, you test cross with a known homozygous recessive. You got to have a little, because every time it gets expressed, you know what it is. There's two ways to be this. So over here, we don't know what these are, but we know what every single one of these are. So if I've got a problem like this on the test, the first thing I'll do is go through and write in every single one that I know. I know every one of those, but I don't know this one. Oh, but I do. Let me cover up this much of this here. Look at their children. Is this one big R or big R little r? Look at their children. What's the only way you could get little r here? Big R, little r. Do you see it? So dad over here has got to be that. Check out the cross. If you don't see it, let me do the cross for you. If you're ever confused by these questions, just do a cross. So this parent we know is little. This parent I'm guessing is big little. It has to be. Because when I work this out, check it out. Most of their babies, oops, I screwed that up, didn't I? Yeah, there we go. get that right now? There we go. So 50% of their babies are going to be little. We've got 2 of them here. They only had 3 babies, so like the probability is not perfect, right? So there's where those came from. So what's this one over here? Big R, or big R, little r? These are the babies from those 2 parents. Is that one big R, or big R, little r? It has to be big R, little r. There's none of their babies could ever have big, because one of the parents was little. See, there's no other way to do it. See how I'm working this out? I've got this sorted. Let's roll over here now. Okay. I've got 2 parents that don't show the characteristic, but one of their babies does. What do both parents have to be? Big, little. Now you're seeing it. Right? The only way for that one to get 2 littles and these guys to not show the characteristic is if both of them have that little r. Okay? Now over here, this baby over here, do we know this one? Could be big, little, right? But it could have also gotten big. We don't know. This one we don't know. It's one of those 2, and there's no way for us to tell. So sometimes that's going to happen, right? Let's see if we can sort the rest out here. Okay. We've got another cross down here. These 2 had this baby. What does this one have to be because they also had that baby? Yep. So it gave the little allele, okay? Over here, we had to give the dominant, so we do know this baby. What's this baby? Yep, big R, little r. Because that one came from there, and this one could only give that, so boom. See how we sorted that out? Like, it was a lot of backwards, like, reverse engineering to get to it, but there's gonna be problems like this. It's kind of fun, actually, if you like things like that. Okay, you gotta sort it, but don't be afraid to just kind of work a couple of different Punnett squares. See which one works. It's just a way of sorting it out. Okay. So we just wrote all the genotypes there. For which individual is it not known? We sorted that out, didn't we? This is the only one that we don't know for certain. We've narrowed it down. Okay. The probability that M and N are going to be photic sneezers. M and N. M and N. So we could work out that probability, right? If I asked you that question, what's the probability these babies will be photic sneezers? You got to do a Punnett square, don't you? With both their parents. Since we know both of their parents, we know them both, we can do that. Remember, the characteristic is big R. 50% of their babies are going to be photic. 50% of their babies are going to be photic sneezers, little r. Okay, so it's 50-50. See how I did that? Super easy. How many generations, right? We just kind of went through the generations. What would be the phenotype ratio if the heterozygous were crossed with N? Okay, well, I've got to work this out in the problem, okay? First of all, let me do this. Let's put N on here. There's N. We know N is big R, little r. So this parent is big R, little r. Now, when I say heterozygous, what does that look like? Big R, little r, or little r, little r? Or big R, big R? Hetero means what? Same, different. Okay, so different would be pretty low. Okay, so we're crossing 2 of the same. And so then I just work this out. Okay, and if we wanted to do phenotype ratios, it would be 3 to 1, or 75% to 25% would be non-photic, and 1% and 25% would be photic seizures. See how we did that? So it's really not that hard, but you've got to be willing to kind of work in reverse. okay, and then play with it a little bit. Be like, I don't know, do a couple Punnett Squares. Was it like this or was it like that, right? If we weren't sure about this, we could have worked this like this, but also worked it with big R, and we would have seen that all the babies would have been big R, right? And would have shown us that it didn't work. Cool? Okay, so that's our review. Go ahead and turn that off here.